All right, I think we're going. Thank you everybody for coming today. This week's speaker is Dr. Jeff Seminoff, and he was on my thesis committee and was such a wonderful influence and mentor. And I'm actually really excited to finally introduce him for this Moss Landing Marine Lab seminar. So Jeff is the leader of the Marine Turtle Ecology and Assessment Program and director of the Stable Isotope Laboratory at NOAA's Southwest Fisheries Science Center in La Jolla, California. Since 1992, Jeff has been involved in ecological research and conservation of sea turtles in the Pacific Ocean. He received his PhD from the University of Arizona in 2000 and was a postdoctoral fellow at the Archikar Center for Sea Turtle Research at the University of Florida from 2000 to 2002. He's authored over 200 peer-reviewed publications, and his research has been featured in magazines, local and national news outlets, as well as on the Discovery Channel, Animal Planet, PBS, and National Geographic Television. So Jeff is really deeply committed to teaching and training young scientists about turtle research and conservation techniques, and he has served on the thesis committees of more than 50 graduate students, including myself. And Jeff's current research uses innovative approaches such as stable isotope analysis, biotelemetry, animal-borne imagery, and aerial surveys to elucidate the life history of sea turtles throughout the world. So I just wanted to also have a couple Zoom reminders for everybody. All audience participants are going to be muted throughout the seminar. Please don't attempt to turn on your video or share your screen during the talk. There will be an opportunity for everyone to ask questions live at the end of the talk, just like a normal seminar. So once Jeff has finished his talk, you can use Zoom's raise hand feature to notify the meeting host that you'd like to ask the speaker a question. And the raise hand feature is located under the participants tab at the top of the Zoom window. And with that, Jeff, take it away.
Thanks, Sharon. Pardon me, there may be some hiccups right now. I have a little bit of an issue with my presentation, but it is a great pleasure to be here today, everybody. And I'll turn my video off in a second. I just wanted to introduce myself. It's kind of weird giving these seminars when you don't have anybody that can give you real-time affirmation or nod or anything, but Gita and Jim and Scott, JD, I think you guys might be out there. Those are some familiar Moss Landing faces that I know. And I just want to say that with Scott Benson and his work, I've had an opportunity to interact with folks at Moss Landing Marine Lab for the last 20 years or so. And I just think that your institution is just such a fantastic program, and I speak so highly of it. And I often comment that master's students that come out of Moss Landing are probably, if not, they're certainly among the most well-prepared students for hitting the job market afterwards. So that's a kudos to the faculty and the whole San Jose State program. So, with that, I just, again, thanks a lot. So let's see here. So the title of my talk, again, I'm going to hiccup a little bit because things don't seem to be operating the way I'm accustomed to. But the title of my talk today is From Molecules to Management, How Stable Isotope Analysis Can Inform Conservation Planning for Sea Turtles. And I'm going to, sorry here, I'm going to turn my video off. Emily had this all figured out and I messed it up for her today. So here we go. So before we get started, I just want to kind of set the stage a little bit and talk about the environmental context of sea turtles and their ecological role. And I think to set that stage, let's just rewind a little bit and recognize that historically sea turtles were once among the most abundant larger vertebrates in our world's oceans. Like this Arabata event in Nancite in Costa Rica shows, incredible numbers of turtles were and continue to be in the world's oceans. And in a photo like this, one of the things that this can really exemplify is the fact that these turtles, sometimes upwards of 400,000 turtles within a couple, 3 or 4 days, will come ashore to lay their eggs on the beach. Not all those eggs hatch. Those eggs serve as prey for a variety of terrestrial predators. They serve as sort of a spark plug for coastal ecosystems. and nutrient flow. And we sort of call this the marine subsidization. It's like the marine subsidy that these turtles bring to terrestrial habitats. So they really are responsible for some pretty amazing aspects of the terrestrial realm as well. Sea turtles are also what we consider regulators of coastal ecosystems. Another term that people have used is ecosystem engineers. And so what I mean by that is by their sheer abundance and them being marine consumers, in this case, the larger photo there is of a green turtle, is that they can actually shape our coastal and offshore ecosystems. And in the case of green turtles, a perfect example is the fact that, you know, they maintain these grazing plots. If you look at the upper right there, that's a grazing plot. It's actually physically altered due to sea turtle predation of turtle grass, in this case, the Thellassia. They eat the seagrass. They sequester, they consume the nitrogen, the phosphorus, the carbon, and everything. They excrete those nutrients. And by doing that, they actually mobilize those nutrients and make them available for the coastal ecosystem much more rapidly than if they had been sequestered in those seagrass blades themselves. So they do kind of act as this nutrient jumpstart for these coastal ecosystems. And the same can be said for loggerheads or hawksbills and their shaping of coastal invertebrate communities and things like that. So you just cannot emphasize the importance of turtles and the ecological role they have. But today, most populations are at a fraction of their former levels due to decades of harvest and fisheries bycatch. And there's a lot of great success stories out there, don't mind you. And in the Eastern Pacific with green turtles, for example, that's one of them. But at the same time, things like Pacific leatherbacks are doing quite poorly. So the statement that most populations are at a fraction of their former levels still, unfortunately, remains the case. But why are they vulnerable to extinction? Let's just kind of talk about that for a second. Well, first of all, they have few eggs and few hatchlings survive. That survivorship is extremely low. It's thought that maybe about 1 in 1,000 eggs survives to adulthood. So a female would have to produce a lot of eggs to reach a replacement rate with something like that. They have delayed sexual maturity, so they take many years to reach adulthood, meaning that they have to traverse all the ocean's threats, the anthropogenic threats out there, just to be able to have the opportunity to reproduce. Females don't reproduce every year, probably every 2 to 3 years, although in low nutrient situations, it can be much longer than that, upwards of 10 years in between nesting seasons. There's many threats to all life stages, from eggs to hatchlings to juveniles to adults. They're all hammered in some way by humans or habitat change. And then lastly, sort of these massive distributions, and a perfect example are the leatherbacks that come to the coast of Monterey. They're originating from the beaches of Papua in Indonesia. So these incredible ranges would require, you know, many different countries to get involved in solutions to protect them. And what I'm going to talk about today to let you know is that stable isotope analysis can actually address a number of the issues that are on the screen right now. And there's some really cool ways that this has been happening, and there's a lot of novel tools, and I'm going to try and touch on some of those today. This is not news to most of us, but I'd say it's sort of the top 5 public enemies for sea turtle bycatch in terms of target species are dorado and shrimp and sharks and tuna and of course swordfish. When various areas have more or less of these various fisheries impacts. I would say here off the coast of California, we probably have some of the safest waters. And certainly if I was a leatherback and I was trying to avoid the fisheries, swordfish fisheries, California would be a great place to be. And I want to introduce kind of sort of 2 topics today. Again, I recognize this is review for a lot of us, but thanks for your patience. We have what's called the Pacific Leatherback Closure Conservation Area, which is that partial red polygon to the north there. And we call this a static closure, meaning that every year for the same period of time, in this case, 15 August to 15 November, it's closed to swordfish fishing. And so leatherbacks, and by the way, Scott Benson's data was directly used to shape the boundaries of this time area closure. And then I'll say for the southern closure, or what we call the Loggerhead closure area, this is focused on loggerhead turtles, obviously, but it's a slightly different type of closure. And we would call this a dynamic closure area. And we say that because it's not fixed through time. It's fixed through space, granted, those are the boundaries, but through time, it is triggered based on El Nino conditions. And historically, it was the official declaration of El Nino that would trigger the closure area, again, to minimize the impact of the the California-based swordfish fishery on loggerhead sea turtles. So in California, focusing on, well, let me step back and say not just in California, but the work that we do at our lab focuses for sea turtles, focuses on abundance and trends, migration and habitat use, feeding ecology, health and contaminants, demography. And when I say demography, I mean things like sex ratio or survivorship or age to maturity, among other parameters. Bycatch reduction and gear trials and monitoring, which we do less compared to other NOAA labs, but that's something that's growing in prominence with what we do. And then finally, sort of a cornerstone of certainly our international work has been the community engagement and citizen science. And what's not on this list are things like genetics and stock structure. And those are not on here because that's more on the side of my counterpart, Dr. Peter Dutton's program at the lab, but we do those things as well. So let's talk about stable isotopes. We'll get into the specifics of it, but just in general, what are the different sorts of questions or concepts that can be addressed when it comes to stable isotope analysis in animal ecology? And well, you'll see the term SIA used throughout. That's simply the acronym for stable isotope analysis. Things like trophic status and niche modeling and diet, those are sort of the traditional sorts of questions that have been addressed with stable isotopes dating back to 30 years ago. And so things like isotope mixing models and ellipse space modeling and SIRE and CYBER, some of the folks on might have heard of those programs, are used to address those sorts of questions. Physiological ecology, looking at metabolism and energetics, is something that's been addressed. You'll hear a lot today about migration, habitat use, and connectivity. And for that, we use things like the isotope tracking technique and taking advantage of the spatial variability in local and sort of global marine isoscapes. You'll also hear today about demography and how we can address questions relating to age at settlement and maturity status. And then community interactions and energy flow are other aspects that I don't necessarily work with. You won't hear a lot about them today, but certainly those are key sort of cornerstones of applications for SIA. As far as the taxa tissues and techniques, everything from butterflies, fish, birds, marine mammals, turtles, variety of taxa. The tissues that are used to analyze are things like feathers or skin or blood or bones, teeth, otoliths, vertebra, scutes from turtles. This is a partial list, mind you. And the techniques, today I'm going to talk almost exclusively about bulk tissue stable isotope analysis. But myself and my colleagues in the community at large, we also employ something called compound-specific isotope analysis, where we're looking at amino acid values of both carbon and nitrogen elements. So I'm here to tell you that stable isotope analysis can provide novel and important insights for questions relating to management and recovery. And I suppose I have to divulge that my underlining of the word "can" there is sort of born out of the insecurity of 20 years of trying to get our agencies to pay attention to this tool to address questions about management. It's a little bit of insecurity, and it's like this, the younger brother saying, yes, we can do it. And I found myself over the last couple decades of trying to, first of all, look for the opportunities to use SIA to address management. And then secondly, trying to in-reach that to the agency. And I have 3 examples today that I kind of want to talk about, and we'll get to those in a little bit. But what are stable isotopes? And this is the most sort of fundamental list that could go on, but for the interest of time, we're not going to spend too much time on this aspect. But isotopes, stable isotopes, are naturally occurring non-radioactive forms of an element, atoms essentially. They contain the same number of protons, but differing numbers of neutrons in their nucleus. And so these differing numbers of neutrons actually have just minor, they create minor differences in atomic weights. And so nitrogen 15 versus nitrogen 14, nitrogen 15 has, its atomic weight is a little bit heavier. And so our animals' metabolism treats atoms of different atomic weights differently. And that is kind of, we'll see examples of that today as we move on. The isotope value of tissue, it's measured with something called isotope ratio mass spectrometry, IRMS, and it's the ratio of heavier to lighter isotopes. I'm using the nitrogen example here, in a tissue relative to a standard. And that's really important, that it's relative to a standard. So you can have negative values, and particularly with carbon, we often have negative values. And isotope values are reported in per mil per thousand. So you can see the various nomenclature there. The isotope values in animal tissues reflect their diet and their habitat and the water they drink, I should say, and they're tracers that can be applied to reconstruct ecological activities and trace ecological processes. And then you'll hear a lot today about how broad spatial trends in isotopic abundance occur, and they relate to latitude, temperature, environmental processes, and habitat type. Okay, so blast from the past, college, graduate level chemistry, periodic table. You can see the red boxes are those elements that have been used in natural sciences. There are pretty much 4 that have been used specifically for sea turtles. By and large, the 2 most common isotopes or elements have been carbon and nitrogen. Hydrogen to a much lesser extent, and sulfur to a much lesser extent. But those are the 4 elements that we look at. We'll talk mostly about carbon and nitrogen today, probably more about the latter, about nitrogen actually. So some important concepts. And I said earlier in that sort of laundry list is that the stable isotope values of an animal tissue reflects its diet and its habitat. And that's true, but there's a conversion factor. And we call that conversion enrichment or discrimination. And what happens is that the animals, a consumer will consume food. It will retain the heavier nitrogen disproportionately in its body tissues, and it's the construction of the tissue matrices, and it purges out nitrogen 14 more so than nitrogen 15. So what happens with that is that there's a stepwise enrichment per trophic level. And the textbooks will tell you it's about a 3 per mil increase per trophic level. That's not always the case. And with sea turtles, it's actually not the case based on some of our empirical research. But still, it provides a framework for us to analyze an animal's tissue sample, get the isotope value, and get some understanding about the trophic level of that animal. So it can be incredibly powerful, especially when you can plug in a particular consumer on this continuum. Another thing to mention, let's just focus on the isoscapes for a moment. So stable isotope values at the base of the food web vary spatially. And typically these values at the base of the food web are what create the isoscape when we develop a map, and you'll see some maps in a moment. But, and we're gonna focus mostly on nitrogen today. So for this slide, I'll just focus on nitrogen. But in general, higher trophic levels result in higher or enriched nitrogen, higher nitrogen values. Coastal prey and coastal predators typically have higher nitrogen values than do offshore predator and prey. And that's actually something similar in carbon, whereas the offshore carbon values, the offshore species have carbon values that are relatively depleted or more negative relative to their counterparts that are in nearshore habitats. So the 7 species of turtles worldwide, it's not a very speciose group. 3 of them, arguably 4, occur in California. We have leatherbacks, as everybody knows on this on the presentation today. Flatbacks, it's an Australian endemic. Loggerheads occur in Southern California. You'll hear me talk about those. Hawksbills, we've had 2 registries of hawksbills in Southern California, so they kind of occur here. And then olive ridley turtles, there's another species that occur here. Their sister species Kemps, and then the green turtle, and their Eastern Pacific variant called the black turtle. So I'm going to give 3 stories today. And excuse me ahead of time, I have 3 yippy dogs in the background, everybody. And so working at home, sometimes dogs get in the way of presentations, but we'll just deal with it. My first story is Western Pacific leatherbacks, focusing on Western Pacific leatherbacks to describe the spatially discrete foraging groups that occur in the region and how isotopes have highlighted the value of the Pacific leatherback conservation time area closure off the US West Coast. The second story I'll tell is with loggerheads in Southern California, clarifying the migratory origins of turtles that come into US waters. And this has provided some great insights for how we manage that southern closure, that loggerhead dynamic closure of Southern California. And then lastly, loggerheads in Pacific Baja. We've used isotopes to define recruitment age, stage duration, and survival at a major bycatch hotspot, and I'll tell that story today too. So, familiar faces here. On the left, Scott and JD. Another colleague of ours from Canada, Mike James. I just simply put this up because I think it's one of the most fantastic leatherback photos we have. Scott could correct me, but I believe it's the largest leatherback we've captured up to this point still, 607 kilograms back in 2007. So with leatherback turtles, a couple of things to point out just to set the stage, because this first portion of the talk will be focused on the species. You know, they nest on tropical beaches. They display natal beach homing, like all turtles, and they forage over vast areas at more high latitudes than most other hard-shelled turtles. They exclusively feed on gelatinous prey. And in terms of the prey, and we have a picture of a chrysora here, is that their prey sort of have these large and rapid changes in abundance, the boom and bust. The turtles are here when jellies are here, then jellies can disappear and the turtles will disappear. But it has been long thought that these jellies are sort of a net sink for carbon and nitrogen and phosphorus, sort of, I guess, a trophic dead end, if you will. I have a question mark there because we're not so sure that's true, because if these leatherbacks are eating jellies, then that's not really a trophic dead end. So I think that paradigm about jellies, as many would agree, needs to be tweaked a little bit. And things like mola mola are another species that certainly takes advantage of jellies for nutrition. But the important thing here is that jellies are assimilating a diversity of nitrogen forms from ingested prey, or the DOM or DIM. And so when leatherbacks eat these jellies, they're almost getting the short circuit to the baseline of a food chain. They're almost eating the very consumers of dissolved organic and inorganic material. So leatherbacks really are a great reflector of what the baseline and what the local stable isotope regime looks like. So the first story I'm going to tell you, I'm going to highlight results from two papers. And I put these papers up here simply to kind of, especially for the students in the room, to talk about how research can be, I guess, iterative and build off of each other. Because the work that we did with my colleague Brian Wallace in 2006 sort of started us scratching our heads. And that curiosity and bewilderment is what directly led into a later paper that Scott and I worked on with some colleagues from Hawaii that use stable isotopes to track leatherbacks in the Pacific. And I'm gonna kind of tell both those stories right now. So for starters, full props to Brian Wallace, a close colleague, probably one of my best friends outside of the turtle world as well. Back for his PhD, he was at Drexel at the time. He's now with an organization called Equilibrium out of Boulder, Colorado. But he was simply looking at the isotope values of two different nesting beaches and the nesting leatherback females that had showed up on those beaches. And so he looked at one data set, and this was a variety of tissue types actually, from St. Croix in the Atlantic and then Playa Grande in the Pacific. And as I mentioned earlier, is that all leatherbacks are essentially obligate gelatinous prey consumers. And so there's not a lot of latitude in there to kind of have different trophic levels. You'd think like a leatherback in the Atlantic versus the leatherback in the Pacific are more or less sort of the same. So they probably have the same trophic level. And if we assume that, then they should probably have around the same nitrogen value, right? Remember that continuum I showed earlier? But that's not what we saw actually. So in this case, and this was a total naive study, and this is 2006, this is way before we had this more acute understanding of isoscapes and the effects of nitrogen cycling processes as we do today. So this was a real sort of eye-opener for us. We're like, whoa. And so when the data finally came back, we saw that there was this huge disparity between the isotope values and the tissues of leatherbacks from Playa Grande versus St. Croix. And granted, the carbon was more or less the same, but nitrogen was just hugely higher in Playa Grande. And if you look at that, like 10 to about 16, that was, as far as the difference in the means of St. Croix and Playa Grande respectively, that would suggest that these animals are almost consuming at two trophic level differences. And certainly that doesn't jive with what we know about their ecology. So what could this be? And this has made us first like kind of look at the isoscape potential. And could there be a baseline value that could be influencing these varying spatial isotopic values? And so our friend Francisco Chavez provided some insights. Here's a map of some of the work that he's done. And it was almost like bing, 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 we found the answer. And this is, to full disclosure, this is it's an isoscape, but it's based on something called N star, which is sort of it measures the linear relationship between phosphate and nitrate, and gives some predictions about what the nitrogen cycling regimes would be in different parts of the ocean. And what we do know is that denitrification drives up nitrogen values substantially, whereas nitrogen fixation drives down nitrogen values within coastal or offshore ecosystems. And so this provided somewhat of a context for us to describe why those turtles from Playa Grande were just so much more enriched in nitrogen than the turtles from St. Croix. But we are also interested, okay, these are nesting beaches, that's great, but what about, again, this is early days, and this work here has not been published yet, but we wanted to kind of just also get a reality check with how do isotope values look at turtles in foraging areas? And so we looked at turtles from Central California. Probably most of these are right around Monterey. We also looked at, with our colleague Mike James from Nova Scotia, looked at the values there. And as expected, we saw the same disparity where turtles from California have a much higher nitrogen value than do turtles from Nova Scotia. The mechanism for this, I should mention, and if you would recall from that isoscape map, that pulse of denitrification didn't make it all the way up to the Central California coast. But there's a process called advection, which is sort of following northbound undercurrents, and those really high nitrogen values and those nitrogen-rich water masses from off Baja and more south actually are advected up into the central coast of California. So we have a mechanism for why this happened. And so this was really sort of an eye-opening, I'll say just for me professionally, of getting a sense of the local impact on stable isotope values of the same species of consumer and how it can change spatially. So graduate, so that's sort of the first paper. That was the Wallace et al. 2006 marine biology. And that made us then think, boy, in the Pacific, there's a great dichotomy or a great disparity of isotope values between the East and West Pacific. And so we thought, let's look at some isotope values back at the nesting beaches at Jamerska Pimenti, which is arguably the largest stronghold for Pacific leatherbacks left in the world today. And so here's what we found. And it was totally surprising to us, is that we saw this this is about 78 turtles. This is just, again, focusing only on nitrogen. And we had this incredible bimodality here. You can see these sort of two groups of animals. It's 3 per mil apart, so it's not the 6 mil between St. Croix and Playa Grande, but still, it's like, why is that happening? Is it trophic, or is it baseline differences? And so those are the interpretations. Like, it could either be two nesting subpopulations feeding in different trophic positions or feeding in different nitrogen cycling regimes. And so if we look at, I don't know if you can see my cursor, but like there's a bunch of foraging areas up here for leatherbacks in the South China Sea, the North Pacific Transitional Zone. Those all have relatively low nitrogen values. If they moved an animal that moves to the Eastern Pacific off the coast of California, granted, remember, it doesn't look like it would be high nitrogen because it's still that sort of lime greenish color off of Monterey Bay. But as I mentioned earlier, there's that process of advection whereby that high nitrogen valued water comes from subsurface currents up into California. And so we thought maybe there'd be a difference there. If we look at it, so here's our map again. Here's our bimodal distribution that you saw on an earlier slide. And we decided to pair stable isotope analysis with satellite telemetry. Photo by Kate Cummings down in the left here, you can see that's an animal that has a satellite tag on it. And the data I'll show you now are going to have some satellite data in it. So it makes sense to show that. So what we did is, this was, 2011, pardon me. We went to Indonesia, collected skin samples, about 13 skin samples. And so you can see these are the samples here. We had about 6 of them that were we went home, we analyzed the samples, about 6 with high nitrogen levels, 7 with low nitrogen levels. And we thought, meanwhile, as we're analyzing the lab, the turtles are out in the ocean moving around, and we're hopeful that we're going to see some sort of trend. And the goal here is we would expect that those high nitrogen turtles that I'll show in blue tracks would have gone to the Eastern Pacific, whereas those red, the turtles with the low nitrogen values, which will be depicted as red tracks, would stay in the Western Pacific. And that's exactly what we saw. And so This is another one of those fall off your chair moments, everybody. You can think about it in theory, but until you actually see your theory come true, it's virtual. And so this is a situation where we realize that not only that nitrogen values of skin provide information on foraging area before nesting, because remember the turtles are bringing that to the nesting beach from their foraging area. And we know that satellite tracking pinpoints areas after nesting. And so when you pair those things together, you see that there's this presence of sort of a near discrete, spatially explicit foraging units. And I say near discrete because there's one turtle that just didn't follow the pattern, that darn turtle. So what does this matter for management? Well, if we rewind to a number of years ago, John Rowe wrote a paper that a number of us were involved with, and that map, your eyes are not off, that is a very fuzzy map, but that's the resolution that it is. And what this does is, if we recognize that turtles in the Eastern Pacific, that go to the Eastern Pacific, are doing that year after year, then let's look at the threats in those areas. And from a sort of density map like this, we can see that the Eastern Pacific is a relatively safe area for these longline fisheries. And I mentioned that, sort of alluded to that with the time area closure map I showed earlier. But if we look at the various foraging areas that turtles could go to, we realize that turtles that go to California truly are probably the safest turtles of any of the bunch. You can see the other 4 areas. There's since been a 5th foraging area located more in the southern Central Pacific. But California turtles or US West Coast turtles presumably should be much better off, or at least face lesser threats than some of those other their counterparts in other habitats. So we figured then, let's figure out how often what's the proportion of animals that would go there. And this is work that Tomo Iguchi helped with a lot. The marginal posterior distributions for nitrogen values are shown here. And we developed a model to figure out the proportion of turtles in each year that would go to the US West Coast. And from 2005, 6, 7, and 2010, we saw that in any given year, about 1/3 to 2/3 of the turtles will come to the US West Coast. And so that really equips us as population modelers to gain a much better sense of, well, first of all, these discrete foraging groups, but secondly, the differential threats from varying impacts to those foraging groups. And then just to tie it sort of back to home, and this is a paper that, and I really have to credit Scott with all this. He's opened the door for us to do all our isotope work on leatherbacks in the North Pacific. But this is a paper that just came out very recently. And if you squint your eyes just right, we know from a Tapilatu paper that the population at the nesting beach is declining at about 6% per year. And of course, this Central California leatherbacks are also declining at a very alarming rate. But if I look at that trend line for the nesting beach, that gray trend line versus leatherbacks, it almost looks like there might be some separation. And it almost looks like California leatherbacks might not be declining at the exact same rate as the overall population. And I say that's very fuzzy language. I don't know, but these are the sorts of questions that we can start to address. And so I look forward in the future to address population modeling of leatherbacks and looking at the various spatially explicit threats for the different discrete foraging groups. So, oh, that's right. One other thing I want to mention is let's go back to that trophic dead end for just a second. And although this is not a thrust of my talk, I did want to give a little bit of airtime to the most traditional use of stable isotope analysis. And that is where you sample a predator and you sample all the putative prey, all the potential prey in the habitat. And then you look at the isotope value similarity between the predator and the various prey species, and you can get some idea of the proportional input of those different prey species into the predator. And there's a black box here. There's some math that happens behind this, but nonetheless, it can be a really effective technique. And this is more or less the technique that's been used to look at trophic status and diet of countless terrestrial and marine species. And in this case, though, much like we had already kind of figured, is that isotopes showed that Chrysora just exorbitantly was the primary food source. And so this is, I guess, if nothing else, it's a ground truth to show that yes, stable isotope analysis can corroborate some of our prior suspicions about diet and predator-prey relationships in marine systems, and probably in terrestrial systems also. So I'm going to move on. Story 2. This story is with North Pacific loggerheads. They're largely pelagic. They eat crustaceans, mollusks, gelatinous prey. They mostly nest in Japan. You can see this, we call them spaghetti plots in the middle there. This is largely George Blass and Denise Parker that helped put these tags on and map these animals. A couple of them make to Baja, but it's an unknown proportion. Most stay in the central North Pacific. And let me just pause for a second and say that until 2014, essentially, loggerheads were really not on the radar of NOAA for addressing a lot of questions in Southern California. But that heat wave changed everything. Wahoo and bluefin and yellow belly sea snakes, wahoo, you name it, all sorts of animals and lots of loggerheads. And Tomo Oguchi led aerial survey effort where we estimated there were more than 50,000 turtles that occurred in the Southern California Bight in 2015-16. So it has really, loggerheads are on our map now. And so that's why this is sort of one of the featured stories for today. But it is important to point out that in that Southern California Bight, loggerheads have been bycaught during El Nino events since 1990, since the observer program started, and of course, prior to that, we would imagine. And the time area closure in that region had been operating under the assumption that those turtles probably were coming up from Baja, loggerheads were, and that's because of proximity. And if you think like spatial parsimony, yeah, the turtles come from Baja, they're not coming from Hawaii. But so if we, and the reason for that is because we knew that Baja was the hotspot for leatherback turtles loggerhead turtles in the Eastern Pacific Ocean. So just by nature, that closeness probably meant that those are the turtles coming up to California. And so we figured, gosh, could stable isotopes help determine origins of loggerheads? Recall that the Pacific coast of Baja had that very high nitrogen value, where there's the Central Pacific not so much. And that was the N-star map, as you can see here. So let's see, like Baja, we'd expect very high nitrogen values of those turtles, low values here. So what does California look like? And that's where we're going with this. So we did look for some empirical data besides the Chavez's map. And we did find something by Lou and Kaplan in 1989 that showed, at depth at least, that the Eastern Pacific had much higher nitrogen values than the Central North Pacific. And that disparity sort of broke down at more shallow depths, but still we imagine that those deep depths and the impact that that has persists in those shallow areas, even despite these data here. So my colleague Cameron Allen led a team of researchers from Southwest Fisheries Science Center to look at stable isotope values from these 3 different areas: Central Pacific, Southern California Bight, Gulf of Ulloa off Baja. And if we looked at carbon, California animals fell right intermediate between those Central North Pacific turtles and the Baja turtles. So we didn't come up with much on that, but again, just one of those moments like, yes, as we saw that actually those California turtles matched up almost exactly with the Central North Pacific turtles. And this meaning that those recent recruits from the Central North Pacific into Baja still are retaining that Central North Pacific isoscape, or that they still reflect that Central North Pacific isoscape. And so this is really, really important for management. And that it forced the agency, NOAA, to think about the fact that, okay, first of all, the turtles are not coming from Baja. Second of all, they're coming from the Central North Pacific. And third is that there's incredible temporal variability in sea surface temperatures and oceanography between Central North Pacific and Baja. And so Michelle Abecassis in 2013 and PLOS wrote a paper with colleagues that sort of showed the preferred habitat of loggerheads in the North Pacific. And you can see how this varies through time. And you can see that probably like that September, like the late August, September timeframe might be a timeframe at which these sort of marine thermal corridors would open up to allow turtles to come into Southern California. Again, it's now we've established this connectivity between Southern California and Central California. So that's the area the agency needs to look at to consider those new sea surface temperatures that would trigger the time area closure. right there. NIMS needs to consider the spatial context for this. And so that is what has happened recently. And I just want to, this is something that Dana Briscoe put together. This is a number of years ago, but it just shows that loggerhead turtles, these are about 14 different animals that are satellite tracked through time, is that they absolutely follow this sort of latitudinal migration of those frontal zones, those SST frontal zones. And as they move across the Pacific, they're moving to the coast of California. And only those animals that are at the right place in time when that thermal corridor opens up are the ones that are going to make it to California. And so that was sort of a really novel context for NOAA to look at when they're developing their time area closures. And so what the agency, my agency, our agency has done since is developed a tool recognizing that fishers don't want to catch loggerhead turtles and they want to know where to avoid, is that this tool, much like something called Turtle Watch in the Central North Pacific, is now focused in Southern California, and it gives fishers an idea of if the closure is in effect or not, and whether or not turtles may or may not be there. So that isotope information was directly relevant for the development of these sorts of tools. So that's my second example. And so as far as the lessons learned so far, the environmental effects of nitrogen cycling, they're conserved up the food chain and expressed in higher order consumers. They provide a mechanism that allows researchers to use the stable isotope tracking method, and they have the power to establish ocean basin scale connectivity. So moving on, I want to talk about studying dead loggerheads. This is the third story to understand the demographics of live turtles. And let's start this story. This is off Baja, that there's mass stranding, mass mortalities as early as 2012. My colleague Hoyt Peckham, who is really responsible for us working down there, sort of sounded the alarm. We recognized something needed to be done. We realized that some of the key things we needed to figure out to learn survivorship is the age at settlement of turtles, how long they stayed in Baja, and the age of maturity, meaning the age at which they left Baja to go back to Japan. And so telemetry could be one tool. The problem is that satellite tags only last 1 or 2 years, and the lifespan of a turtle is up to 60 years. So we needed to develop a different technique. And that's where looking at sea turtle humerus bones came into play. This is the shoulder bone of a turtle. And we can track individual movements over multiple years using these bones. And there's two approaches that we use. One is skeletal chronology. The second is stable isotope analysis. And I want to give huge props to Callie Turner-Tomaszewicz. She's a postdoc at our lab. She's been a fantastic colleague. She's a total rock star with publications and written a lot of methods papers for isotope analysis and skeletal chronology and the use of the two together. That's where the humerus bone is on the animals. So using isotopes to study sea turtle movements is much like dendrochronology. Each year of growth in the humerus bone is like a year of growth in a tree rings, essentially. And so you can look at a humerus bone, you prep it the right way, you can get some idea of the age of that animal. And a turtle like this that wound up on the beach in 2009, you can back calculate both its year, its size, and its age. And you do some modeling to figure out how many of the years were resorbed in those innermost, the cortical portion of the bone to get an actual age of the animal. That gives you the time. Then what about location? And that's where stable isotopes come into play. And so again, back to the isoscape, we know that turtles are coming from the oceanic environment into nearshore habitats. The isotope values are going to be different from the two. And with nitrogen, we expect the nearshore values to be much higher than the offshore values. So kind of we can model the transition of animals from habitat A to habitat B. And so we take that humerus bone, we slice it like a loaf of bread. We had the one half that we use for skeletal chronology, the other half we use for stable isotope analysis. We put notches in it so we can match up the two. We're able to identify the areas, those annular growth rings. We then sample in those annual growth rings to sort of get a sense of when those animals transition between habitat A to habitat B. And this gives you some idea. And this turtle was a 9-year-old turtle that settled into Baja. And this gives you an idea, just another sort of perspective of the drilling that happens in these bones to get an idea of the age at which these animals transition habitats. We use something called a Carpenter Systems Series 2 microsampling robot. You put the bone on there, it samples it for you, you collect the dust, you analyze it, and you get the insights So the important thing to ground truth this is having some known location samples so that we can say, yes, that habitat A truly does have these values and habitat B does have these values. So when we look at the growth rings, we can really pinpoint where the animals came from. And we do have known location samples. And this is all through Callie's leadership. And we really showed that there are two very different isotopic profiles from Central North Pacific versus Baja turtles. It's about 5 per mil difference in the mean values. And when you think about like a transition between the two, a threshold value, around 15 per mil would be the time. If an animal has an annual growth rate that's 15 per mil, that's from Baja, essentially. So you can do it with a few different animals. I'll show you here. So here's one turtle, CC 25 June 1001. This turtle transitioned at about 9 years old. Another turtle, 10 years old. Another turtle, 18 years old. And if we go back to Archie Carr's Lost Years, which suggested turtles stay in the open ocean for only a few years, certainly this is changing that paradigm. But if we do this with a bunch of turtles and looking at that transition from one habitat to another, this is 45 turtles and about 38 of them, 37 of them actually we had data to be able to pinpoint the age at transition. What we realize is something that we also didn't expect is that there's two pulses of recruitment, sort of a varied life history strategy within this population. You have these small settlers or these small shifters that come in at about 7 years old, and then larger shifters that come in around 16 years old. And we believe this is simply because some were lucky and at the right time at the right place, and other turtles, it took them an extra 9 years to be at the right time in the right place. But so in terms of survivorship, if we understand now like how old these animals are when they're coming, and if we look at the size of all the turtles in this habitat and recognizing that these animals are anywhere from 30 to 90 centimeters, and we know the age of those turtles, we know that in that habitat, those turtles live there for upwards of about 20 years before they swim back to Japan to nest. And again, when they go back to Japan, they never come back. They stay in the Western Pacific after they leave Baja. And so what this does, again, is it shows us sort of alternate habitat use patterns, whereas at the top, it could be turtles that kind of go to the Central Pacific after leaving the nesting beach. These are the small animals. Then they stay there for some period of time, probably like the large shifters, and then they get to Baja, and then they mature and go back to Japan for the rest of their lives. Other animals, like the small shifters, probably spend less time in the Central North Pacific and move almost straight to the Eastern Pacific. And then the third group was probably the largest group, are turtles that actually never make it to the Eastern Pacific. They stay in the Western Pacific. But how does this portray for survivorship? And so if we know there's about 43,000 loggerheads that occur in the region. We know that the annual mortality rate is around 11%, or at least it was with Volker Koch's work. The animals that come straight to Baja as youngsters and spend the full 20 years there, their survivorship rate for the stage is only about 10%. However, if an animal shows up in Baja at a larger size, that survivorship increases substantially, upwards of about 30%. So you can see the implications of these alternate habitat use patterns in terms of stage survivorship, and you can have this huge range. And this is all just information that became available because of stable isotope analysis. So you really can learn an incredible amount from a single bone. That's sort of the take home from this. And it's not just bones. So stable isotope analysis of serially deposited tissues has been done for a variety of taxa, a variety of tissues. Humeri, squid beaks, baleen, sperm whale teeth and pinniped teeth, fish otoliths, shark vertebrae. They've all been used to more or less do exactly what I just showed you for that loggerhead story. So for those of you that might be thinking of projects or trying to answer a particular question about residency time, if you have an animal that has a serially deposited tissue, there might be some great promise for the stable isotope analysis technique. So for my sort of take home, the wrap up here is that when spatial heterogeneity in marine isoscapes is present, like in the Pacific versus the Atlantic, the Brian Wallace story I told you, SIA can depict migratory connectivity on broad scales and inform population assessments and MPA planning, like the value of the leatherback conservation area. When combined with other research tools, stable isotope analysis has the power to elucidate the amount of time individuals stay in specific habitats and helps define demographic parameters like survivorship as well as susceptibility to threats like bottom set gillnets and drift gillnets and longlines in Baja. And then sort of what I've been trying to sort of convince everybody is that existing and novel stabilized isotope analysis approaches are being increasingly used to inform management and conservation of species. And I've given you 3 examples where those have the 2 Loggerhead and the 1 leatherback story have absolutely been integrated into NOAA's thinking about things. And just as a take home in my last sort of few seconds here is I just want to talk about novel. And this is something I just added because it's really cool. It's where my lab is focusing much of our effort. It's a developing story with my colleague Seth Newsom, who's just become an invaluable colleague, and one of his graduate students, Laura Page, is on whose committee I also served. And it was addressing the question is, can stable hydrogen analysis determine reproductive status of green turtles? So I'm throwing a little curveball here. It's a different species. This is maybe a 4th story, but it's a partial story, so I didn't include it up front. But just a background, Laura's master's research focused on hydrogen isotope discrimination. That is, what is the difference between stable hydrogen values between the predators and their prey? She worked with captive turtles at Cayman Turtle Center. It used to be called the Cayman Turtle Farm. It's a wide range, including juveniles and adults. They were reared around a constant diet, so sort of a perfect experimental setting. And we had somewhat unexpected results. And that is that stable hydrogen, the values were vastly different in juveniles versus adults. And we attribute this to varying levels of lipids in the blood, probably because adults have circulating lipids for vitellogenesis and reproduction, whereas juveniles don't have that. But we're just starting to explore that more deeply now and focusing on a population of Hawaiian green turtles in the wild. This was great in captivity, but we want to look at it in the wild. And if indeed stable isotope analysis can start to address questions about reproductive status, that could be an absolute game changer in terms of population modeling. So that's certainly a growth area, not just for our lab, but for other labs. And it's another example of how isotopes can address questions relating to management. So with that, I will say thanks for your patience. And I sure appreciate the attention, although I can't see anybody. And I will stop right there and go back to my video and again say thanks to everybody at Moss Landing Marine Lab.
Well, thank you very much, Jeff. That was a great talk. I already see that there's a question. So it looks like Jim has a question for you.
Hi, Jeff.
Hey, Jim.
So my question is has nothing to do with anything you talked about, which is with climate change and changes potentially in productivity throughout the world's oceans because of it, could stable isotopes be used to sort of actually look at changes in the oceanographic conditions over time, or is stable isotopes too variable to be able to pick up maybe what might be more subtle changes?
No, I think it could, Jim. We know like denitrification really skyrockets in areas with robust oxygen minimum zones. And so that certainly could at the mercy of climate change as temperatures and acidity changes. And so I could see in the long scale, and granted, this is on like massive global or ocean basin scales, but with Francisco's sort of efforts with NEN-STAR, I could see something where we could, at least for oceanographic processes, start to map how they change. How that portrays an animal ecology, I'm not quite sure yet.
Cool. All right. Thanks. Good seeing you.
You too.
Good talk.
And Geeta, nice to see you too.
Nice to see you. Yeah. We have another question already. I see Lauren has her hand up.
Hi, Jeff. That was a really interesting talk. I had a question about what your role as a scientist working for NOAA Fisheries is in actually changing those policies and the ultimate management decisions that are made with all of this data that you're helping to collect. Like, what is your role in that other than you collect the data, you interpret it, and then do you hand it off to other folks, or are you involved in that decision-making process?
Yeah, well, it's the latter. We're definitely involved. And I can't comment for how scientists are involved in policy decisions in NOAA as far as an agency around the nation. But I will say that, and it's probably because of the fact that our regional office, which is largely based out of Long Beach, have some people that really recognize the value of science. And we have an incredible relationship with those folks. And so folks like Penna Revuelos and Chris Yates and Tina Fey and Dan Lawson are all people I work really closely with. And Tina was deeply involved with that, the Loggerhead closure and trying to reassess how to figure out that the trigger for when or not to open it. And to give you like sort of inside the room is that what would happen is we'd develop these studies. We'd let them know that we're doing this. They really sort of appreciate that. And then it is almost iterative where they ask us to analyze information. They'll look at the data and then maybe, can you look at it a little bit different way? Or can we look at sea surface temperature in this quadrant versus this quadrant? So it's been a very, for me as a scientist in NOAA, has been extremely participatory. And I would say probably that Loggerhead Southern Closure is like the crown jewel example of how the agency managers and the agency scientists have kind of worked hand in hand.
Are there any other questions for Jeff? Oh, there is a hand. So it looks like Alex has a question for you.
Hi, Jeffrey. Great talk. Thank you so much. I had a question. You mentioned that both not loggerheads, leatherbacks and ocean sunfish eat primarily gelatinous prey. And these are both like really large bony animals that are eating like this really nutrient poor diet. So how does that work? Is it just like a very large amount of this prey, or why do they specialize in this way?
So two things that I can think of. One, Alex, is that they eat a ton of it. And secondly, is they focus on those body parts that are most rich in nutrition and lipids. So they're focusing on the gonads of those animals. And as Jim can attest to more than me, but there's times when you can be offshore, like on the Sheila B with our twin otter flying ahead and JD's driving the boat, and it's really sort of leatherbacky looking water, and you'll see a bunch of these sort of bells of jellies with no gonads or oral arms or anything. And it's because the leatherbacks came up and ate all that stuff. And there's incredible video on that. So there's been a lot of effort looking at sort of the energetics of how these marine megavores can actually sustain on these. And it really, it's about volume and picking what you want to eat.
All right, last call for questions. Or we let them off the hook. But it looks like that's it for today. Thank you very much. That was a great talk.
Yeah, I sure appreciate it, guys. It's great to see you, Gita and Sharon and Jim, and I appreciate everybody's time and attention today. And if you ever have questions about isotopes, my email, somebody can find it for you, and I'm happy to help out anytime. So thanks so much.
Thank you. Have a great weekend.